in the microsporogenesis, the broadly, as I already told you, that the microsporogenesis is nothing but the formation of a microspores. Nothing but the pollen grains. We already know that the anthers, which are acting as a carriers for the pollens. Nothing but the pollens are the male reproductive structure, which are essential parts where they through the abiotic factor or through the biotic factor or by any means they shift from they will move from the one plant to the another flower by in search of the stigma, style as well as a ovary. So here the formation of a pollen grain is the development of the immature anther till to the formation of a pollen. Okay, now, so further pollen developments we'll study in the male gametophyte. Okay, now, so till to the formation of a microspore, that is what we call it as a pollen. That is we are going to understand in the microsporogenesis. So nothing but the development of a microspores from microsporocytes. That is what we call it as the microsporogenesis. And usually what happens, the formation of a microsporogenesis doesn't, that is how we have seen in this particular slide, it is not looks like that. Okay, now the formation of the development of a microsporogenesis, which usually occurs by a oblong or quadrangular or irregular quadrangular clump of cells. Those are again a meristematic cells. We'll show you the image of that, how they are, and you all are already familiar with that. I would like to give some information, some ideas about that. Okay, now. So initially the microsporogenesis occurs by the immature anther. That immature anther, which just looks like an irregular quadrangular or oblong in shape, which is completely made up of meristematic cells, where that particular meristematic cells which are surrounded by the epidermis, nothing but the immature or a young or which is said as the actual microspore forming initial cells will be having only the meristematic cells which are compactly arranged, which is surrounded by the epidermis. Fine, and these what happens during the course of a development, what happens, the meristematic cells initially divides to form enlarged cells. So all the meristematic, let us take this what a clump of cells, and they are completely filled up with the different type of meristematic cells. So where in the four corners, as I already told you that they are oblong or in a quadrangular in shape, there in the four corners of the immature anther, there you can see the formation, these meristematic cell activity defines, and there we are going to see the enlarged cells which are quite different than the surrounding or neighbor meristematic cells, and which are how they are different. They are morphologically bigger than the neighbor meristematic cells, though they are meristematic cells itself which are quite bigger than the neighboring meristematic cells, as well as and they will be having a dense cytoplasm and a very conspicuous nucleus, and that's what we call it as the archisporial cells. These archisporial cells will be called as the initiators or the initial cells for the formation of the microspores and other microsporogenesis parts of the microsporangia. These what to call these microspore these archisporial cells which are quite different in a morphologically, as well as though they are having the capacity to divide, because the meristematic cells are all the meristematic cells are having the capacity to divide because they are perceiving the totipotency mechanism or the totipotency principle, where they divide and they may form a particular structure, and that archisporial cells, they are quite different in a morphologically, and that's already given in the fourth statement that is there, that we can observe it when you take the immature anther, and you can stay if you stain it and in the laboratory you are going to see then these archisporial cells which are quite highly or greatly stained than the neighboring meristematic cells, and there you can see the active very conspicuous nucleus and are dense filled with the cytoplasm, where these archisporial cells, broadly when we talk about the particular that you all can
Sir.
can you all able to see, yeah, now can you all able to see the image, this one?
Yes, sir.
Yeah, okay, fine. And
Yes,
here you can
yes,
see
sir.
that these are the four. Okay, these are the four microsporangias, and I mean this nothing but which carrying the microspores, right? So initially these are not. I mean, we are not going to see such morphologically a peculiar structure like this. So the formation of these four chambers or the lacules is just because of the activities rendered by the archisporial cells. Fine. And we'll go to the next one. And you can see that there are only 4 archisporial cells in this particular image. Right. So the number of archisporial cells usually which are confined to only 4 in most of the angiosperms, whereas in some of the what we call the specialized in some of the angiosperms where you can see only one archisporial cell which is forming the microspores. And in Ophiopogon is again a type of is again a plant which contains a number of the archisporial cells which are arranged in the form of a plate. So the archisporial cells are also can be used in different types of the embryological studies, where most of the the plants which confine themselves to have only 4 archisporial cells which are arranged in the 4 lobes of the quadrangular immature anther. Where in the Borevia we can see only one archisporial cell, and in the Ophiogoson we are going to see the varied number of archisporial cells which are arranged in the form of a plate. These archisporial cells, what happens, what actually the function of these archisporial cells, and why we have to stress more on these, because the young or immature anther which we have, which we say it as they contain the meristematic cells. When these meristematic cells which will divide and form a different structure, where these archisporial cells are also having the meristematic property, and they divide and form the 2 different types of structures. They are morphologically quite different, and that is what one is a primary parietal cell, another one is a primary sporogenous cell. Nothing but the primary parietal cell, nothing but these are they divide the cells which have been formed after the division, the meristematic activity of the archisporial cells, which are going to form towards the epidermis. And nothing but these are involved in the formation of a wall layers, different types of a wall layers, and we'll tell you what are those wall layers which have been formed by the primary parietal cell. Excuse me. It may be, except epidermis, all the walls, it may be endothelium, middle lamella, and parts of the tapetum, all are going to be formed by these primary parietal cells. And the primary sporogenous cells, these are the cells which are involved in the formation of the microspore mother cell as well as the microspore. Nothing but the pollen active dividing pollen grains are going to be formed by the primary sporogenous. Is that clear, guys? Parietal cell. And when we talk about these two, when the archisporial cells are able to divide in 2 different planes, I mean, in what happens, the primary parietal cells, the previous slide, as I already told you that the primary parietal cells, which initially divides both in anticlinal as well as a periclinal division. So what happens, because of the anticlinal as well as the periclinal division, I mean, you people have remember what you mean of anticlinal. What happens in the anticlinal division? Nothing but the dividing cells which are perpendicular to the plane of axis. Like nothing but this sort of plane of axis, and the division occurs perpendicular to the plane of this one. So what we are going to see, we are going to see the 2 neighboring cells which are arranged one side by to the another cell. Where in case of the periclinal division, what happens? The the division occurs parallel to the plane of axis. Nothing but we are going to see if this what a cell, it is going to divide in this pattern, and where we are going to see the arrangement of 2 divided cells one above the other. That is what the periclinal division. So here the primary parietal cell which will undergo a repeated many number of anticlinal as well as a periclinal division. So why? Because they are exclusively needed for the production of internal structure. That's what I already told you, that they are going to form, they are responsible for the formation of a different wall layers. So usually what is the role of any wall? It may be the epidermis, it may be the endothelium, it may be any type of a wall. Main role is to protect the internal parts. Where in case of a primary sporogenous cells, that's nothing but a sporogenous cells which will not take much of the divisions. They may divide for quite times, and they may switch off their division capacity once they find the number of microspores or pollen grains are found inside of the lacule or a cavity. Okay, and these particular parietal cells which were giving, they divide and they give rise to several layers, and they are as I already told you that they are going to form the wall of the anther. I always remember, guys, these parietal cells which are not involved in the formation of epidermis, because epidermis is already available. Epidermis is already there in the immature anther. These epidermal cells, as and when there is an expansion in the size of the anther, there we are going to see these epidermal cells are going to divide again, and they are going to fill up the area as there is an expansion, as there is an increase in the size of the anther. Their epidermal cells are itself themselves having the capacity to divide, and they are going to extend their protective layer as the dermal tissue, or they are going to form the epidermis itself. And here, in terms of a parietal cell, these parietal cells or a primary parietal cells are nowhere directly related to the formation of epidermis. They are going to form only the protective layers which are just situated below the epidermis until to the central part, nothing but till these sporogenous cells are going to form. So these from epidermis till to 3, 4, 5, or 6 or 7 layers of the walls are going to be formed by the parietal cells. And next, these sporogenous cells, the second one is they divide few times, quite a few times, and giving rise to microspores or the pollen mother cells or a microspore mother cell. Don't get confused always with MMC and Barita. MMC nothing but preferably it has been used for megaspore mother cell. So to avoid the confusion, and sometimes even for microspore mother cell also we can mention it as MMC. Sorry guys. So to avoid the confusion, you can make it as PMC, pollen mother cell. There, in case of when we talk about megasporogenesis, there you can use MMC for megaspore mother cell. Fine, megaspore mother cell for the female reproductive structure, and the microspore mother cell. That's what we call it as a PMC, pollen mother cell for the male reproductive structure. Where what happens below the epidermis, as I already told you that we are going to see the formation of very next to the epidermis, we are going to see the wall formation from the archisporial or a primary parietal cell. That is what the endothecium. So this endothecium sometimes which is going to be only unilayered, or sometimes it may be 2-layered or 3-layered, which is just below the epidermis. For example, this is what the outer structure of an anther, and which is having a unilayered epidermis. Below that, below the epidermis, we are going to see the endothecium, which is quite often 1 to 3 layered. Where what happens, this particular endothecium will lose its capacity, nothing but it is going to become degenerated as the sporogenous cells will mature to form a mature pollen mother cells, or when we are going to get the mature microspores or active pollen grains, these endothecium which is situated below the what we call the epidermis, which is going to become degenerated. So and it is going to degenerate, and it becomes like fibrous structure, and which is get attached to the epidermis, which is just forming as a dry coat on the sporogenous cavity. And we'll show you the image of that. Fine. And next to the what we call next to the endothecium, we are going to see the formation of 1 to 3 layered middle lamella. So this middle lamella, which is again because of the activity rendered by the primary parietal cells, where first epidermis, next to that the endothecium. The endothecium is going to be formed by the primary parietal cells, and next to the endothecium, you are going to see the middle lamella. And guys, in most of the textbooks, the endothecium which has been given as only unilayered. But very recently, we have come to know that there are 2 to more often unilayered endothecium you are going to see. But sometimes you are going to see the 2 or 3 layered endothecium also. Next to the endothecium, you are going to see the 3-layered, or again here I mentioned it as 1 to 3 layered, but usually unilayered middle lamella we have not seen it anywhere. But 1 to 3 layered middle lamella we are going to see just below the endothecium. That is what we call it as a middle layer between the endothecium and inner wall. That's what internal wall is, the tapetum. So this tapetum, which is acting as a primary nourishing wall to the developing microspore or a pollen grains. So the primary parietal cell, which is involved in the formation of a wall, and there again morphologically quite changeable, quite changing wall cells are there. One is the endothecium, followed by the middle lamella, followed by the tapetum. Tapetum, which is the innermost wall, which is acting as the protective to the microspores, as well as which is provides nourishment to the microspores for the development. And these, what we call, when these sporogenous cells, when the pollen grains starts, divide, they undergo division, the meiotic division. Usually what happens, the reproductive structures undergo meiotic division. So during the meiotic divisions in the microspores, these wall layers, it may be anyone, it may be the endothecium, or it may be the middle lamella, it may be the tapetum, will get crushed up. Why they are going to crush up? Because if they are completely protected by these wall layers, these microspores cannot come outside of the anther. So they have to come out. So for that, they are going to weak, they are going to be crushed by different forces, and finally these microspores, or mature microspores, are going to become out, and these pollen grains will be in search of the female reproductive characteristics for the reproduction. And what happens during the microspore mother cell, or I mentioned as MMC, when they undergo the meiotic division, we already know that the meiotic division, which is the reductional cell division, nothing but where the diploid cell will become a haploid cells, and these haploid cells, nothing but these pollen grains, which will usually contain the the what we call the pollen, or a male gamete usually contains the haploid haploid set of chromosomes, and when they find their suitable haploid ovule, and they are going to fuse with that, and they are going to, when there is a compatibility, they are going to see the fertilization. So here what happens in the microsporangia, where we are going to see the four nuclei which are arranged tetrahedral. So that the pollen will show you the pollen how they are arranged in tetrahedral array. So it just looks like a diamond. There 1, 2, 3, and 4. The tetrahedral arrangement of the nuclei we are going to see in the microspore. So these microspore are nothing but the mature microspores. So once they start, when they undergo the meiotic division, we are going to see the four haploid microspores which are arranged in the tetrahedral manner. So once the tetrahedral manner arrangement of microspores are going to be seen, the tapetum is going to be the tapetum is going to be degenerated. Nothing but the tapetum is going to lose its existence. Then the microspores are pollen grains which are going to be dry up, because till the maturation, till the growth, they need to get some of the nourishment, and it may be in the form of a moisture, it may be in the form of a nutrient, it may be in the form of anything they are going to be received from the tapetum. So when the tapetum loses its existence, then there is no such nourishing layer. Nothing but these microspores are matured enough so that they can be transferred, they can be expelled from that particular sporangia, and they can go in search of the female gametophyte. So these dry up just like microspores. We already know that pollen grains are just like a dust particles. When they get hydrated, and they are going to start germination of a pollen tube. Right, such thing we are going to see, and that is what the formation, the development of a male gametophyte, as well as the reproduction part, it may be the pollination as well as the fertilization at the time. We'll explain those things at the time. So today we'll confine myself to this microsporogenesis. Finally, what happens when the anther becomes a mature anthers? Then what happens? The partition walls are going to be broke down, and through the what we call the partition walls, where we can see that the dehiscence of an anther, where we are going to see the formation or expelling of the well-developed, well-mature pollen grains. These microspores or pollen grains are going to be dehisced through the what we call slits or splits and corrective, where we are going to see the openings of these what we call the chambers, where what happens during the development of a pollen grains. We already know that this pollen, which will usually will be having the two protective layer. One is the exine, another one is the intine. Intine, which is internal protective tissue, and as well as the exine, which is the outer protective tissue. We'll talk about the pollen during the pollen grains, where the tapetal cells are also not only involved in playing the major role in the nutrition, they are also sometimes acting as the they may provide their role in the formation of the exine too. Exine, which is outer one. So because the tapetal cells, which are very near to the sporogenous cells, and these during the degeneration, these walls may get adhered to the pollen grains, and they may what we call settle on the exine or outer layer of a pollen, and they may form they may act as a the substitute external substitute for the pollen as an exine. And this is what the schematic representation of the development and the formation of the anther. And guys, you can also able to see this first one in the A image. There you can see that oblong structure, which is having only the epidermis. At the center, you can see only the meristematic cells. So these these meristematic cells, you can see there is an, as and when there is a shift from A, B, C, D, E, F, G, there you can see there is an increase in the size of the microsporangia, right? So this you can see at the four corners, we are going to see initially the formation of archisporial cells. So in the first in the corner, in the four corners, we are going to see the formation of archisporial cells, and you can see that there are quite different they are quite distinguishable than their neighboring or their adjoining meristematic cells. Can you all able to see the difference here? They are quite
Yes, sir.
sustained more. Yeah. So just see these archisporial cells which will divide, and where you can see the initially archisporial cells which will divide either anticlinally or periclinally. So there we are going to see the formation of, let us stick on ourselves that they are undergoing periclinal division. I think, but this particular archisporial cell which is divide periclinally, and they may form, one is parietal cell, another one is a sporogenous cell. Always remember that the parietal cell which are divides and which are going to form towards the epidermis, where the sporogenous cells which are going to divide and form towards the center. Nothing but they divides in this direction, and the parietal cells which are divides towards the epidermis. So as there is a shift in the, as there is a development, as well as, as there is a division in the what we call the archisporial cells, there you can see there is a shift in the shape of an anther. You can see there is a, from oblong to there we can see finally just like a butterfly structure of the microsporangia. So finally we are going to see when the parietal cells start dividing, where we can see formation of an endothecium and the middle lamella, I mean middle layer, as well as a tapetum. And these three, endothecium, middle layer, as well as tapetum, all these three together will be called as the anther wall. Epidermis, which is the protective outermost wall of the anther, where the endothecium, middle layer, and the tapetum, these three together will be called as the anther wall, which is directly protecting the internal porogenous cells. So when they start dividing, and there we are going to see the formation of a, there is again what we call shift in they divides, and finally they are going to form a peculiar structure of the endothecium, and this endothecium is going to be completely protecting the sporogenous tissue. Middle layer is going to form next to the endothecium, and finally at the innermost layer, that is what the tapetum, and this particular tapetum which will be nourishing the microspores. And here we can see that this sort of vascular bundle connectivity, nothing but though it is again connected to the plant itself, so it is the vascular bundle, nothing but the phloem as well as the even the xylem, which are yet still connected to this. Finally, we are going to see the structure like this. So when the microspore mother cells are there, you can see the structure arrangement like this. So when the microspore mother cell will undergo meiotic division, they are going to form the tetrahedral pollen, tetrahedral microspore, and at the time you are going to see the microspore like this. And you can see even there is a break in the slits are going to be formed, and through the slits we are going to see the expelling of the microspores. Is that clear? Just I wanted to uh, uh, give some abstract by taking this particular diagram. So we already know that, as I already, don't get confused with the remaining circular structures. Just observe only what I'm uttering the terminologies. Okay, na? So this is what a first cell. This is what undifferentiated anther. The undifferentiated anther, which divides, nothing but which will be peculiarly will be having only two structures. One is the epidermis, outermost one. Then next one is the meristematic cell. Though the remaining meristematic cells are not at all directly needed, we are not going to study during the microsporogenesis. So I'm sticking myself only to the archisporium. Nothing but undifferentiated anther. There we are going to see the two important structure. One is the epidermis. Another one is the another one is archisporium. This particular epidermis, during the course of a development, they divides, and they are going to form only the epidermis, nothing but which is acting as outer protective layer to the particular, the whole anther. That this archisporium, which will divide and form, archisporium divides and forms the two structures. There you can see the primary parietal cell as well as the primary sporogenous cell. These primary parietal cells, these are responsible for the formation of, there suddenly a single parietal cell will not directly form the different wall layers or anther walls. Initially they undergo the division and forms a number of, the comfortable number of the parietal cells are going to be formed by the primary parietal cell. Nothing but the primary parietal cell which will undergo the division, and by the division they are going to form some required number of cells. Accordingly, we are going to see the secondary parietal cells. So these secondary, in those cells, let us make it as, let us call it as a secondary parietal cell. In that, some of the secondary parietal cells which will divide and form the endothecium. Got it? So as I already told you that in most of the textbook they have given it as only a single layered endothecium, which is nothing but outermost anther one. So I have mentioned it as only one. Next, some of the secondary parietal cells which will divide and form the middle layer. Middle layer may be sometimes one or two or three layered. So we are going to see the upper middle layer are going to be formed by the secondary parietal cells 1 and 2. Fine. Nothing but endothecium. If there are three layers of a middle layer below the first and the second, below the endothecium, first and the second middle layer cells are going to be formed by the first group of a secondary parietal cells. And in the secondary parietal cells of a second group, the lower or a third layer or a innermost middle layer are going to be formed by the some secondary parietal cells, whereas some of the cells are responsible for the formation of an innermost cell, that is what? The tapetum. So can you all able to see this one? So primary parietal cells which divides and form the two a quite number of secondary parietal cells, and these secondary parietal cells, out of those, for example, on ನಾಲ್ಕು ಇದೆ ಅಂದ್ರೆ two are responsible for the formation of endothecium layer. The remaining two only one is going to these two which will responsible for the formation of the 1 and 2 middle layers. One layer of a middle layer or a middle lamella, and the second layer of a middle lamella. And in some other clump of a secondary parietal cells, they are going to see the innermost middle lamella. Nothing but if it is three layered, the third middle lamella, the lower middle lamella will be formed by the different group of secondary parietal cells, whereas the innermost cells, nothing but the layer that is what a tapetum, is also formed by the primary as well as the consecutively formed secondary parietal cells. Now coming to the another big part of the archisporium, that is what the primary sporogenous cells. This primary sporogenous cells which will initially form the secondary sporogenous cells. These secondary sporogenous cells will divide and form the microspore mother cell or a pollen mother cell, and these pollen mother cell or a microspore mother cell, they undergo the meiotic division and they are going to form the haploid tetrahedral microspore. So this is what the abstract of the today's class what you have discussed. Is that clear? Which is explaining about the anther wall as well as the epidermis. You can see the outermost one is the epidermis, and you can see the endothecium which is a unilayered, and you can see the bilayered middle lamella, and we can see the microspore which are microspore mothers which are intactly arranged to each other, and the innermost one is the tapetum. There you can see the star grains are there, and in the conspicuous a big nucleus is there which is quite distinguishable than the microspore mother cell as well as the other anther walls. These tapetum which will be acting as a nourishment as well as sometimes they are also responsible, they are add-on substitute for the exoine molecule during the pollen formation. So this is what about my today's class. Is there any doubt, guys?
No, sir.
No, sir.
Okay, then
No,
if there
sir.
are no doubts, fine. If there are no doubts, we'll end the session now.